everyone, it's Maddie here to talk about the final lab. Congratulations, you made it to this point. I'm first going to talk about the preparation of your specimen. So from previous labs, you have had many specimens that you prepared. So the first step will be to pick one of those specimens and we are going to cut it to shape it because this lab you're going to be examining the specimen under a microscope. So this is the apparatus we're going to be using to cut the specimen. If we look in here, we can see that I have already placed a specimen in here. These pieces, these clamps, if you raise them and lower them, you can control the forward and backward movement. And once you have this set and tight, lower them to make sure everything's in place. This hood is an emergency stop, so if you have any issues at any time, quickly lift this back up so it will not start moving until this is shut. The on and off switch, also make sure that is switched to on when you're ready to start cutting. This blade here is very fragile, so make sure that when we go to cut the specimen, we slowly lower to cut rather than a quick drop. So when we're ready to go and we've got everything in place, we will shut the hood. We'll make sure that we've got our light on and we've got the flush switched on. And then this emergency stop, when it's pushed in, nothing will happen. If I pull it out one time, you'll see liquids start to spit out inside the apparatus. That's just to keep the specimen cool when we're going to cut. And then if I pull this out one more time, the blade will start moving. I'm going to grab this lever and slowly pull. All right. Push the emergency stop, and then we can lift the hood. And we can see that our specimen's been cut. All right, once you guys are done with the sectioning portion of the lab, you can look at your specimen, you can remove it from the apparatus with some pliers. You'll know if it's small enough because we're gonna come to the mounting section, and these are the mounting cups we'll be using. So if your specimen can fit in here, then it's perfect size. All right, so if you look at your specimen and notice some of the edges are a little too sharp, you can use a deburring tool if you would like to remove the edges. So some notes of importance about the fume hood. So anybody who is actually using the fume hood and will be using the liquids inside, make sure you have gloves on. If you're not working with the fume hood, however, you do not need the gloves. As low as you can manage to operate the fume hood, keep it that low. If you need to raise it a little bit, do not raise it above this yellow line. So to make our mount, we are going to be using powder, and we'll be using liquid. So it's important to follow the directions on the mounting powder, which we can see right out here that it says we will be using 2 parts powder to 1 part liquid. So we'll be using these little measuring cups, and we can see that we've got lines on the side. So I'll start with the powder, and I will fill it up to the second mark, which I'm not going to do right now. Then I'll come to the liquid. It's very important when you're using the liquid to pour in the cup sideways rather than vertically, or else you'll have clumps when you're going to dump the liquid in to the measuring cup. Once you have the amount of powder and liquid measured out in the cups, you'll grab one of these paper cups and you will dump the powder and liquid. It doesn't matter which order, just make sure it all gets into this cup. And then you'll grab a popsicle stick and you'll just start stirring for 30 seconds until you see kind of a paste forming in the paper cup. We'll come over and grab our mounting cup and our specimen, and we will place the specimen into the bottom of the cup, or if you would prefer to analyze your specimen from a side view, you can use I don't know if you can see it, one of these little tools and attach it into your specimen like so, so that when you're placing it into the mounting cup, you would place it in vertically and go from there. So again, I'll come back to the fume hood, I'll place my specimen, I will grab the mounting liquid powder mix and I will dump it into the mounting cup and fill it till it's approximately 3/4 of the way full. Any smaller, it's going to be extremely hard to manage your specimen later in the lab. Once I have the mounting material placed into the mounting cup, I will leave it under the fume hood for about 30 minutes so that everything can get settled inside there. Very important when you're using the fume hood, keep everything under here as best as you can because we don't want smells coming out into the lab area. And now I will show you, so once we have the mounting liquid powder mix in here and it's been set for about 30 minutes, we will see a harder liquid powder mix that's no longer a paste. And to remove it from the mounting cup, we're just gonna pop off this bottom, if I can get it. Like so. And I will just push the new mount out, and this is what it should look like. Okay, and very important, after you remove your new mount, write your name and the sample type so that you don't mix them up when you're doing the lab. If you need more mounting material, because you are mounting 2 specimens, you can just repeat the steps and create that for your second mount.
Hi, it's Daniel again. So we'll go over grinding and polishing step. First, these are 2 examples of poorly mounted specimen. This specimen is too thin. You'll have a difficult time grinding it. And this specimen is not level, which is as a result of not having a level cup when it was mounted. So to operate the grinding machines, you first want to make sure that the appropriate water valve is in the open position. You will adjust the stream of water till it's either a slow stream or a dripping pace. For this grinder, you'll start it up by turning the time dial to infinity all the way over here. You will adjust the RPM so it's between 240 and 300 RPM. You'll make sure the emergency stop button is all the way out. Now that we have the grinder running, we'll start at 180 grit sandpaper. You can find the sandpaper discs in those boxes. Have one dedicated sandpaper grinder and another grinder for each of the polishing steps. To go between the sandpaper grits, remove the metal ring and place the disc of sandpaper down. So when grinding a specimen, make sure that you're keeping it at a constant orientation. Have the water running, and for about 2 to 10 minutes, make sure you try and use the surface of the sandpaper evenly. Also, make sure all the other grinders are covered at this step to avoid cross-contamination. All right, well, we're done there. All right, once you have your specimen, have all the grinding lines oriented in the same direction, move on to the next grinding level and orient the lines 90 degrees to the rotation of the disc. All right, for the polishing step, we'll be using 3 different granularities of alumina powder suspended in water. And so for each of the other 3 grinders, we will take the appropriate alumina size. And you'll use this in conjunction with a grinder loaded with a nap cloth instead of sandpaper. You'll get water running. and you will start this up. Once you have the wheel running and the water stream going at an appropriate rate, you will take the alumina powder and just have it, just spray it gently underneath the, while the sample's being polished. The alumina powder is a lot finer than the available sandpaper, and it'll bring the polish and it'll help polish the surface further. All right, once you're done with the final polishing step, you should see no more scratches present on the surface of your specimen. And at this point, it's very important that you don't touch the surface of the specimen at all, as that could potentially leave some markings that will show up on the microscopic analysis. So once you're done polishing, place the sample specimen side up inside the sink. You will rinse it off with the water bottle. And then after you rinse off the specimen, use the air nozzle to rinse off the water. With that removed, you will take the alcohol bottle, which will be labeled here, and you will rinse that off and let it air dry for about 30 seconds. Once you have it dry, you will take the appropriate etchant for the material property of the specimen, and you'll apply a few drops to the etchant surface, to the material surface. You'll let it sit for 20 to 30 seconds. It's important that it doesn't sit too long or too short, as this will cause problems further down the line. Once you've let it sit in the etchant, you will rinse it off with water and then rinse it off with alcohol and let that air dry for about 30 seconds. All right, once you have your specimens ready, between the 2 specimens you've prepared and 3 already prepared specimens of your choice, you will analyze them using the microscope. So first, open the PaxCam software. Once the software is open, you can see what the microscope is looking at. So to adjust the major resolution, you will turn the knob with the various eyepieces laid in. And so start at the lowest resolution and gradually move inwards. The fine adjustment knob will help enhance the resolution at a specific zoom quality. And so the green bar in the top left of the software window is a representation of how good the resolution is. So you just adjust that with the fine tuning knob. Hit autocorrect if necessary to account for exposure. And there's also another knob here that you can use to move around your specimen. So, as you can see on the computer, this is one of the pre-prepared specimen. Notice the fine grain structure and the absence of any scratches or other defects present on the surface. Here's a specimen that was not as well prepared. And as you can see, there are a variety of scratches that will make analyzing this specimen more difficult. If these scratches are prevalent, you would want to go back to either the grinding or polishing step, re-etch the specimen, and repeat that until you have a satisfactory image quality.